In this video, I'm going to talk about the Dihybrid Cross and the Punnet square. I'm going to show you how to use the Punnet square to prove the 9:3 to 3 to 1 ratio. So this is the ratio for the Dihybrid Cross. Before we go further, let us talk about the Dihybrid Cross. What is the Dihybrid Cross? What do you understand by the word Dihybrid Cross? So Dihybrid cross just simply means the type of crossing in which we focus on two traits, on two different traits. In a monohybrid cross, we focus on a single trait. But in dihybrid cross, we focus on two different traits. From the prefix di, it simply means two. The prefix mono simply means one. So in dihybrid cross, we focus on two different traits. You need to understand that. Trihybrid cross, we focus on three different traits. So what are traits? When you hear the term trait, just simply means the variation in the characteristics of an organism. So the variations in the characteristics of an organism, it is called trait. So for example, we talk about roundness. We can talk about the color. We can also talk about the shape. So if you say blue, yellow seeds, blue flowers, wrinkled seeds, so those are traits. Okay? So dihybrid cross, they conform to this ratio, 9:3 to 3 to 1, which was even proved by Gregor Mendel. But there are some types of dihybrid cross doesn't conform to this. So some they don't conform to this ratio. And what are the reasons behind that? So the reasons behind that is because of gene regulation, and also we can talk about the polygenic control, and the other reason is that gene linkage. So we're going to talk about that in the next videos. So imagine that you've been given this parental genotype. So this is the male parental genotype, and it has been crossed with a female parental genotype of similar nature. And then they tell you to find or to prove the 9:3 to 3 to 1 ratio. How can you do that? Using the Punnet square. The first thing that you have to do, you need to self-cross these genotypes or these alleles. So we are going to get the genotype for the male parent, which is R, Y, like that. And then we are going to self-cross it. So we're going to do like that. So let us now determine the F1 genotype. So you've got R, uppercase R and uppercase Y, uppercase R, lowercase y, lowercase r, uppercase y, lowercase r, lowercase y. So these are the alleles, or in other words, we can say these are the gametes. So what are the gametes? So when you hear the term gametes, what comes in your mind? So gametes simply means the sex cells. So all the sex cells, they're called gametes. So we've got the sperm and also the ova, which is the egg. So these are the gametes in human beings. So the alleles carried by the gametes are the one that you've determined here. So this one, that, and that. So this process of crossing over occurs during the pacting stage of prophase 1 in meiosis, which is meiosis 1. So this is where the genotype, or in other words, the alleles from the mother and the father, they are mixed, or they are interchanged. The genetic material is interchanged. So as you can see here, we've got separation of alleles. So alleles are separated. So this is under the law of segregation. Mendele's law of segregation. It states that alleles, they segregate in meiosis. So now let us see how we can use the Punnet square to find the 9:3 to 3:1 ratio. And then take a note that the genotype for the male parent and the female parent is the same. So the gametes are going to be the same. I mean, the alleles in the gametes are going to be the same. As you can see, we need to put the alleles or the gametes for the father or for the male parent on top. So as you can see here, these they are representing for the male parent, and these alleles this side they are representing for the female parent. So when you start crossing RY and RY, it's going to give you this genotype. If you do the same for RY and RY, it's going to give you that. Okay, so what you have to understand here is that capital R presenting roundness, so it's for round, and then small letter r is representing wrinkled, then capital letter Y presenting yellow, small letter y, it is representing what? Green. So when Y is dominant, the seed is going to be green. When Y is dominant, or which means I can say when green is dominant, the seed is going to be green. When yellow is dominant, the seed is going to be yellow. So now let us look at what we have here. Let's try to analyze this. So let us try to focus on this one. We've got capital R, which means round is dominant, and the small letter r, which means wrinkled is recessive. It's not going to show in the phenotype. And then we've got YY. So yellow is also dominant. So for this one, it's going to be round and yellow. That's why you can see this one is round and yellow. For the other one, I've got capital R and small letter r. Capital R is for round. So round, it is dominant. And then wrinkled, it is recessive. It's not going to show in the what? It's not going to show in the phenotype. And then we've got capital letter Y, which is yellow. So when you try to count the round yellow that we have, you find that we've got this 1, 2, 3, 4, 5, 6, 7, 8, and 9. So we've got 9 round yellow. When you count our round green that we have, we've got this 1, 2, 3. And then when we count our round wrinkled, our yellow wrinkled that we have, so yellow wrinkled, we've got 1, 2, 3. Then our green wrinkled, we've got only 1, which is that. So this is the ratio. So we can say that this crossing, or this dihybrid crossing, was conforming to the Mendelian 9:3 to 3:1 ratio. So this is what you can do. This is how you can use the Punnet square to prove this ratio. If you've been given a question in test, choose the Punnet square. Just remember that the gametes for the male parents, they should be placed on top. And then for the female parents, on the left-hand side. So that's what you have to do. As simple as that. So guys, this is what I got for you. Thank you for watching. Don't forget to subscribe to my YouTube channel for more videos.